Yeah, welcome to our webinar series. And I'm Rene. I'm from the measurement committee. And today we are talking about the importance of parts. So this webinar series is kind of divided in small sections, so it's easy to follow. And I will do the first two sections, and then Tanya, also from the measurement committee, will continue for the third section. We will allow questions for all three parts. So after every part, I will stop and we will open up for questions. And I will start with slides, but in the end, I will also show some like examples which showcase what I meant with these slides, kind of. Okay, can you all hear me loud and clearly? Just one last feedback.
Yes.
Is it okay? Perfect. And okay, so as I said, the first part of it today is the importance of parts. And I would like to give you an overview what I will present in the first 15 minutes. And this is about what means parts and what are parts in engineering in other engineering disciplines, and what does parts mean in synthetic biology. And the second part is, why is standardization important for SynBio? And can we learn something from other engineering disciplines on that? And then we came to the invention of the first standard for SynBio, or like the first standard assembly technique, the Bioreassembly assembly. Although it's kind of not outdated, but invented a long time ago, and now teams are using different standards. I would like to come back to that, explain it a bit, and then in the last part, I would come to the new standard, or the standard which came to iGEM 1 year ago, and that's the Type 2S fighter rig standard, and so on. Okay, let's start with parts in common engineering. So imagine you would like to build an airplane, or your smartphone, your complex circuit boards in electrical engineering, and the parts which are used in these engineering efforts would be as undefined, I would say, than we did biological science in the past. So these efforts, or like these successes, were just so successful and possible because the people who engineered these objects used standardized parts. And these parts are well defined. You know perfectly the function of a transistor or something like that on an electric board. You know perfectly the materials which are used, and you know the properties of these materials. And therefore you can recombine all these parts and use them also for completely different other engineering efforts. And these parts are tested in simulations, but also in real life. You can also imagine all the rocket testing, what SpaceX, for example, is doing. So all these parts are defined, and you know exactly what they are doing. Now the question is, do we have the same situation in synthetic biology or in biology in general? And of course, we would like to have that exactly in that way, that we think about engineering biology in that way, that we have different levels of abstraction, it's called. So DNA is the lowest level of abstraction, and you could call systems or multicellular organization as the highest level of abstraction. And this is exactly what synthetic biology tries to get from these engineering principles, to have standardized parts and use abstraction to make the engineering process easier. You would also not, for example, to give another example from informatics, or computer science, you wouldn't do a Twitter post or something like that in 0 and 1s. And that's the same for DNA. So actually people try to program DNA on the 0 and 1 level when you look on the DNA level, but now you move up the levels of abstraction, and now you can do something like a Twitter post without typing 0 and 1s, right? And parts could be defined by functions, so promoters, RBS, one coding sequence or a terminator, that would be a part in biology, right? And that's something which is kind of at the heart of iGEM, to use parts with one certain defined function and to collect data for them. And why is that important for iGEM and for SynBio? The biggest advantage of having these small defined parts is that we can exchange them with all the other iGEM teams and all the other SynBio labs, and you can combine them in any manner or order, and you can characterize these parts in a collective manner. And for that reason, if many iGEM teams and labs over the world would use the same part in different conditions, in different chassis, in different ways, you can have a collective effort to get to a point where we are in common engineering, that we exactly know the function of, for example, one certain promoter of one certain ribosome binding site or something like that. And that's kind of the dream, that we as the iGEM community do the collective effort by creating but also characterizing these parts. And just to come back to the invention or the first assembly method, and that was the biobrick assembly. I will not go like totally into that now, but I would like you to understand the basic principle of it. And the basic principle is that you have parts on different plasmids. For example, the top left 1B0034, that's the RBS, and so on. And you have all of these parts, no matter which part it is, flanked by the exact 4 restriction enzyme cutting sites. And therefore, you can cut all of them you would like to combine with its same 2 restriction. Oh no, not the same 2, but the restriction cutting sites which are flanking, and then you can make use of that. You can combine restriction cutting sites from 2 different enzymes, which create a mixed ligation site, and therefore you have a combined composite part with 2 parts or biobricks used. So as I said, I won't go into detail for that method because not many teams are using it anymore. But if you want to know more about it, there's a link here, or 2 links which explain it on the one hand in the theory, and also on the other hand how it works in the lab. So if you would like to use biobrick assembly, you can use these 2 links to get more into detail for that method. And based on that standard, on this biobrick assembly, teams created since 2004, since iGEM started, many parts which are now in the registry of parts, and we come to that more in the second session, where I also show you how to add a part and how it should be. And all these parts were sent in a physical form in the past. So the teams really sent the physical tube of DNA to the iGEM headquarters, and you were able to order your parts you would like to have from other teams, but you also get this distribution kit, which I showed here, where you have 6 or 7 times 384 wells of biobricks, which you get every year, basically, besides this year because of the special situation. But this was the idea in the past. So, but now teams don't have to send the physical form of DNA anymore. So now it's just about filling in the DNA sequence into the form, and all the parts are synthesized afterwards by company. So you are not forced anymore to send the physical DNA. But all these parts are still in that old kind of standard, or you have to follow that standard at least, or since last year you can also use the new standard, but all the parts in the registry follow that standard, so you can get them and directly use them for combination. The new standard, which is allowed since last year, is the phytobrick standard. And just before I go deeply into that, there will be another webinar next week about golden gate assembly and phytobricks, modular cloning. Just if you're interested in that, there will be a super detailed introduction next week on that. But just to give you a first introduction for that, I would like to explain a little bit about that standard here. So in that standard, you just have one restriction site, and these restriction sites work a little bit different than other restriction sites because they cut outside their recognition sequence. That's the BSA1, and therefore you have these N base pairs, and with that you can define different positions and different part positions, and therefore you have standardized overhangs for that cutting site, and also standardized illegal enzymes, which you shouldn't have in your part. There shouldn't be any of these sequences, and that's also kind of part of the standard, that you avoid with the restriction sites in your parts which are part of the standard. And you can find these lists of diffusion sites online on the iGEM website, but I just included them here. And I like this figure a little bit more. So originally this standard was developed for plants, because that's why it's called phytobricks, but you also have a bacterial version of it. So you have defined overhangs for promoters on left and right and so on, and defined overhangs for RBS and so on, and then you can combine them in one pot. And that's the difference. That's the difference to the Biobrick assembly. In Biobrick, it's a little bit slow because you combine part by part in one cloning round, and to build up one plasmid, or like one transcription unit which expresses you one protein, you need several rounds of cloning because you just add one part. And in contrast to that, this new standard, this type 2S cloning, allows you to combine all these elements, promoters, RBS, coding region, terminator, and so on, in one pot. And then after one cloning round, you have combined all these in one assembly. As I said, if you're interested in that, we would like to come back or invite you to the webinar next week and explain a lot more about that, the technique, and how you can design your project around these phyto bricks. So now I would like to show a showcase, kind of what I saw as a judge 2 years ago, and that's not like an exact example, but I just created kind of a dummy plasmid map for that. And what I saw as a judge 2 years ago, that many teams kind of forgot of this kind of part thinking, that you are dividing your whole plasmid in parts, and that you can switch out parts to have like this modular designs. And a lot of teams just, I don't know, got a plasmid from the fridge of their lab where they're working and just switched out somehow with some cloning technique the gene of interest they would like to express, or even stacked the gene of interest behind each other without thinking about to have this as a part. And then in the end, because it's an iGEM requirement, they just brought it back to the iGEM format in the standard vector. And I mean, you could do that, but this is actually not the philosophy of this part, thinking and synthetic biology and standardization. And that's one of the examples. So many people use these PAT16S vectors for protein expression, and that's fine. So people who are just interested in expressing one protein and purifying them, they are probably happy about that that vector. But yeah, so this is kind of also sometimes unpractical if you want to find out which circuit or which, I don't know, metabolic construct works for you. And I would suggest every team, to really go into some modular or part-based cloning, no matter if it's Golden Gate, modular cloning, whatever, or any other cloning technique. But I really like teams which have the kind of part thinking in their mind, that you have like for every part that's now Golden Gate assembly or modular cloning assembly, and that has also the advantage that if you design your whole project in that way, that you have like promoter, RBS, and also terminators and your gene of interest, if you have that separate and it doesn't work, what is quite likely in many projects that something doesn't work, then you have so many options to shift your project or to shift the way you approach your problem. Then if you just use some of the vectors you have in your fridge, which your old group or something like that used in the past, without this part thinking, and in that case you can, for example, switch out the promoters to a more highly more expression or a weaker expression or something like that, or exchange the RBS because of that kind of part thinking, and you have everything divided. And also the characterization, that's kind of the second part, which I come to in a minute, is easier in that case. Okay, that's the first part. That's not very practical, I know, but next week when we come to the phytobrick session or the Golden Gate MOOC session, then we will show you a lot more how to use that cloning technique. for your project. Okay, now let's have questions. You can ask in the Q&A section questions, but I guess you could also raise your hand and I would allow you to talk. But both is fine. You can put it in the Q&A or raise your hand. I don't see any questions. Do the other measurement committee members see any questions?
Yeah, Rene, there was one question. Someone was asking, how are we going to get parts if we don't get the plate this year?
Aha, yeah. Actually, this is also included in my second section. So I will answer this question in the second section. So wait for it, just 2 minutes.
And there's another question here in the Q&A about what is the best free software for engineering DNA parts?
So I wouldn't say there's any best software, and many softwares are in general not free. But on the one hand, there's benchling, that's always free. That's an online-based software, which is where you have all your files online. That's free, and iGEM is collaborating with them. Then there's Snapgen, and that's in general not free, but iGEM has or iGEM student have Snapgen and Genius free to use for any iGEM student. So you can get a free license for 1 year. For example, this is Genius. I like Genius because they sponsored me kind of since my first iGEM year 2016, and that's why, because I'm just used to it. But that's just my opinion. But yeah, as I said, Benchling, Snapgen, Genius, all of these 3 are free for every iGEM team, and then you should just try and find out what works best for your team and for yourself. So I wouldn't say there's a huge disadvantage for one of them. Could you give a particular good example of a team thinking about their project in a modular path-based mindset? That's actually also part of my second part. I will answer that in a minute. How do you learn all these things very intensively with just internet resources if you are a beginner? That's a good question. Actually, these webinars are one way of learning it, right? And we will also upload these webinars so you can watch them several times. If this is too fast for you, that's totally okay, and that's also not bad. And we will also provide you with some links to really learn the basic stuff. And in the next weeks for the webinar, there will be a lot of basic questions, basic techniques, which I explained by our committee. And yeah, you can follow this webinar, and I think this will help you a bit. Alexis?
Yeah, I'm just going to add here. So all the presentations with the software that we're going to show for this series of webinars, whether it's being demoed by someone who uses Benchling or Snapgen or Genius, you will always have another video online showing the exact same demonstration with another software. So someone is asking in the questions, if you want to try them all right now, you could download them all, try them, and then stick to one, or you can just follow some webinars and do the same thing as one of the presenters is doing.
Okay, yeah. Okay, there are two open questions. Do you mean it's better to have every part on a separate plasmid than combining them through any technique? I think this is kind of the one of the bases of synthetic biology, that you have parts separate which have a certain function and then combine them by any technique. So that, in my opinion, it's I don't know if the word better is the right word, but this is a proper engineering way of approaching a problem. Okay, what do you think about synthetic biology EDX MOOC? It's a good or not? Is it good enough to understand all these things? I don't have an answer to that. Can anyone else from the committee answer that?
So yeah, the edX MOOC is good material. But if you follow the webinars that we're giving this summer, it should give you the basis that you would require to build your circuits in the lab for iGEM. So you don't have to, but if you want some complementary info, I invite you to follow this MOOC. It's a good one.
Yeah. Are the software licenses only valid through the duration of 2020 iGEM cycle, or do they last indefinitely? Actually, most of the softwares last for the entire year of iGEM, so not just until the Giant Jamboree, but not indefinitely. But these companies are very kind. If you have, for example, an iGEM project which you would like to continue for some months, I have the experience, if you approach them and say, okay, in my iGEM project, we started that and we have to finish that. Can we have some the license for some months? So I can't promise that, but I know that they are very open to that, to help you out. But in case of benchling, you wouldn't be dependent on that if you want to make it really sure that you can use the software indefinitely.
And I may add, someone was asking earlier, Snapgen has a version that's always available, which is called Snapgen Viewer, yeah. Which doesn't have the other functions. You can't edit, but you can always visualize your plasmids with Snapgen Viewer. Benchling is always online. And there's also another repo called SquidBio, where you can store your DNA online, and they have good visualizers.
Yeah. Okay, the last question I see here is, how can you know in advance about the biological trade-off if you have a lot of parts? I don't know if I understand the question correctly. And I try to answer it somehow. And if I didn't answer the question well, then just ask again in a different way, perhaps. So of course you cannot know what the exact or the perfect part combination is, but in case of setting up your project in a part fashion, or in a way of dividing everything in parts, it's more easily to adapt to problems or to expression levels which are not appropriate for your purpose, and that's why it has an advantage. Of course, you can't know the trade-offs for any combinations or something like that. Did this answer your question? Like the energy resource that are needed that can lead to bad expression rates later. Actually, there are some resources on burden. That's something some iGEM teams also have investigated last year. So, for example, different promoters or different parts have different burden, or as you called it, need different energy resource of the cell. And actually, there is a way of measuring these burdens, And of course, it would be nice if we would have a burden value for all these parts. That's not existing yet, but if iGEM would be willing to continue on that, yeah, it would be nice to have all these burden values for all these parts, right? But unfortunately, you're right. We can't foresee every burden which is caused by all our parts we introduce into the chassis. Okay, should we continue to the next part, or are there any more questions? In case of a promoter, so I can clone on one vector to regulate protein X, then clone it to my plasmid. I'm not sure if I get the question correctly.
So I might try to guess here. You might use one backbone for cloning, which is just a way of reproducing your plasmid, and then you could have another backbone for expression.
Yeah, that's possible, of course, yeah. And also, if you can, what's quite common for these parts is that you have them in a kind of cloning vector for propagating them and also for cloning them. And in the end, for the expression, you combine them in a different, or clone them in a different vector, kind of. I hope this answers your question.
And just to make things clear for everyone, we will have a web page that goes with this webinar that already has some material and that we will update with the answers that we're giving today.
Okay, should we start to the next, go to the next? Any more questions? If we use a type of assembly in a part fashion and find a problem, why would the fact that it was assembled in a part fashion solve the trial and error process? It doesn't solve the problem and the trial error if you are not relying on kind of, I don't know, super proper modeling, but it's easier if you have. So what I do for my projects now, for example, as I'm used to that part thinking and part cloning, I create these parts or these proteins I would like to express, or these enzymes, whatever, as a part. And afterwards, I combine these parts with a lot of regulatory elements, so a lot of promoters and so on at the same time. So I'm able to kind of do a lot of different constructs and expression levels at the same time due to the fact that I use that part type of creating my plasmids. And in the end, I test all of them at the same time. And I would say that I'm a lot faster with that way of working because I'm kind of not stuck to one backbone or not stuck to one promoter or something like that. I can tinker around with all these different expression levels and ways of creating my constructs in a very, very fast way, and you can also automate that a lot easier. Is it possible to take out the part that you think is not working and suitable with a different one while keeping the rest of the construct same? For some cloning techniques, that's of course possible, but the EcoFlex kit is it's a good example, but in that case you can't take out one part. Then you would just assemble the construct from scratch, kind of, and that's not even slower than doing it from how to say it. That's not slower than exchanging one not working part by just using different parts, putting them in a one part assembly reaction, and then it's fine. Yeah, it is like the EcoFlex kit stepwise, right? Yeah, that's correct.